Okay, the today's discussion is, as I have said, is the topic which is monoclonal antibodies. Okay, so when we talk about monoclonal antibodies, we are essentially talking about antibodies which are produced from a specific clone of what you call B lymphocytes, okay, or plasma cells. So the antibodies which has been produced from a single clone of B lymphocytes, produced from a single clone of B lymphocytes, okay, will be called as, or will be termed as, monoclonal antibodies. Okay, so when we talk about single clone of B lymphocytes, what exactly we are trying to say? So what exactly happens is, whenever an antigen enters into the system, we have already spoken many a times, a particular B lymphocyte, not a particular B lymphocyte, multiple different B lymphocyte will try to understand the structure of the antigen on the basis of the different what we call receptors present on the surface of the B lymphocytes. Okay, so let us say clone 1, this is clone 1 and this is clone 2. Okay, so two different B lymphocytes. So out of that, what has happened is a particular kind of interaction was there with this particular B lymphocyte. It has been seen that this particular epitope, or let us say epitope 1, if we call it as epitope 1, this is epitope 1, and let's say that this is epitope 2. Okay, so what has happened? Epitope 1 is interacting with the clone C2 receptor. On the contrary, the epitope 2, now this is the antigen, remember, this is the antigen. Okay, so it could be any antigen. Most of the time this could be a bacterial antigen or a fungal pathogen as well. So when these antigens will enter into the system, what it will do is it will stimulate the B lymphocytes, and they will come together, and there will be an antigen receptor interaction. Now a receptor which is present on the surface of the B lymphocytes. So what exactly happens is, after the binding, a process will start which is known as clonal selection. Okay, so what will happen? A clonal selection will be initiated. Clonal selection process will be initiated, and eventually each of these C2 and C1 cells will differentiate in multiple times. Okay, so after the clonal selection will take place, what will happen? These certain clones which are very effective against these particular epitopes which is there on the surface of the antigen will undergo propagation, and so many more clones will be formed. Okay, so many C2 will be there, like this, C2 and C2. Okay, here again C1 for a different epitope. Remember, for a different epitope, C1 and C1 will be formed. Now what will be the function of all this? They will all start producing antibodies. Okay, what they will do? They'll start producing antibodies. Okay, they'll all start producing antibodies, but these antibodies are specific to a different kind of epitope. So we shall call these antibodies, let us say, anti-E1 antibody. Okay, now this is an anti-E1 antibody, and this will be what? What could be the nomenclature of this? Anti-E2 antibody. Okay, understood? So what exactly is happening? Whenever the antigen has entered into the system, it has eventually stimulated what? The production of B lymphocytes. Now these B lymphocytes are having certain cell membrane receptor on the surface of it, which interacts with the different epitopes of the antigen. Usually an antigen is a multi-epitope entity. An antigen is what? A multi-epitope entity. Okay? On the surface of a antigen, most of the time there are multiple epitopes. Okay? Unless and until you have made into a subunit vaccine. Okay? The rest of the time what will happen? The normal antigens, they are having multi-epitopes. So for example, epitope E1 and E2. Now they will interact with two different kinds of B lymphocytes only. Remember, these are all B lymphocytes. Okay? B lymphocytes. So two different clones of B lymphocytes, clone 1 and clone 2. Now clone 2 is effective against the epitope 1. On the contrary, the clone 1 is effective against the epitope 2. Now, after the binding of the antigen and the receptor, clonal selection will take place. Remember, clonal selection will take place, and eventually the propagation of each and every clone will be there. Now, each and every B lymphocyte clone, what ultimately they will do? They will produce antibodies. Now, this C2 clone will only produce anti-E1 antibody. On the contrary, the C1 clones will be producing anti-E2 antibodies. Now, each of these antibodies, E1 and E2, each of these antibodies, E1 and E2, will be termed as what? Will be termed as what? Monoclonal antibodies. Okay? So, write it down. Monoclonal antibodies. MOAB. Okay? MOAB. Okay? That means what? Monoclonal antibodies. Although their specificities are different towards different epitopes. Okay? So, what will happen is, so what will happen is, two different monoclonal antibodies will be present, but because it is both the antibodies are present on the single serum sample, isn't it? Therefore, they will make what? They will make into a thing called polyclonal serum. Okay? So what they will make into? They will make into something called polyclonal serum. Okay? So we shall call them as polyclonal serum, which is present in your body. Okay? Polyclonal serum, we shall call it. Okay? So you understood? So what exactly is a monoclonal antibody? Monoclonal antibody is an antibody. Remember this. Monoclonal antibodies is an antibody which has been produced from a single clone of B lymphocyte. Okay? Now, polyclonal antibody is what? Polyclonal antibody is the mixture of all these monoclones, that is, which is present inside the serum. So, whenever we are talking about the serum, we are essentially talking about what? We are essentially talking about polyclonal serum. Okay? Now this serum, which I usually prefer to draw in this way, okay, what it would be? It would be a polyclonal serum. Okay? Now this is the serum that you have isolated in a test tube, and in the test tube this is nothing but a polyclonal serum. Whenever you isolate serum from your patient, so this is nothing but a polyclonal serum. Okay? So you understood what exactly is monoclonal antibody? Okay?